Function of the digestive system is to break down food that you consume to release nutrients, which then can be absorbed into your bloodstream to be transported to cells in your body. Digestion of the food can be classified as either mechanical or chemical. Mechanical digestion involves physically breaking the food into smaller pieces. It begins in mouth, where food is broken down with help of teeth, and carries on in stomach, small and large intestine, where the muscles continue to break the food. Chemical digestion, on the other hand, involves breaking down of small chunks of food into simpler nutrients with help of various enzymes. It begins with an enzyme called salivary amylase, which initiates breakdown of carbohydrates in the mouth. Lingual lipase is an enzyme which initiates lipid digestion in the stomach. And pepsin is an enzyme which initiates breakdown of protein in the stomach. In the small intestine, carbohydrate, lipid, and protein digestion is completed with help of pancreatic amylase, pancreatic lipase, and trypsin, respectively. Now, the easiest way to learn digestive system is by grouping organs into two categories. Organs that make up alimentary canal, that includes mouth, esophagus, stomach, small and large intestine, and accessory digestive organs, including salivary glands, liver, gallbladder, and pancreas. The entrance to the mouth are lips, which are very vascular, hence the reason they are red. They keep the food inside of your mouth. Cheeks make up side walls of the mouth. While their outer covering is skin, their inner covering is mucous membrane. In between skin and mucous membrane, you will find connective tissue and baconator muscles, which help you chew the food. Inside of the mouth or oral cavity, you will find gums and teeth, which begin mechanical digestion of the food by physically breaking large chunks of food into smaller ones. At the roof of the mouth, you will find hard and soft palate. Hard palate, located anteriorly or in front, serves as wall which separates nasal cavity from the oral cavity. It also helps the tongue push the food. Hard palate is hard because it is composed of maxillary and pontine bones. Soft palate is located posterior to the hard palate, meaning it is in the back of hard palate and is mainly composed of skeletal muscles. Soft structure hanging down from the soft palate is called uvula, and together with soft palate prevents food from entering the nasal cavity during swallowing. Uvula also contributes to snoring. At the floor of the oral cavity, you will find tongue. Top and sides of the tongue are covered with fungiform papillae, which contain taste buds, and filiform papillae, which contain touch receptors. So your tongue plays an important role determining whether food is edible or not based on how it tastes, and it also determines texture of the food. Lingual glands are also located in the tongue, and they secrete mucus and lingual lipase, enzyme which initiates chemical digestion of lipids in the stomach. There are three types of salivary glands in the mouth. Parotid salivary glands, located between skin and masseter muscle near the ears. It secretes saliva into the mouth through parotid ducts. Sublingual glands lie below the tongue and secrete saliva into the mouth through lesser sublingual ducts. Submandibular glands are at the floor of the mouth and secrete saliva into the mouth through submandibular ducts. Now, saliva secreted by each type of salivary gland has slightly different consistency due to cellular makeup of each gland. For example, parotid glands secrete watery saliva, whereas submandibular glands secrete thicker saliva due to the presence of mucus-secreting cells. And sublingual glands secrete the thickest saliva and least amount of salivary amylase. From digestive point of view, saliva contains salivary amylase, enzyme which initiates carbohydrate digestion in the mouth. It also contains salivary mucus, which helps to lubricate the food and facilitate movement of the food, making it easy to swallow and move down. What's also present in saliva is the bicarbonate and phosphate ions, which contribute to the alkaline pH of saliva, specifically pH between 6.35 and 6.85. Saliva also contains immunoglobulin A and enzyme called lysozyme. Both act as antimicrobial agents. Saliva also contains epidermal growth factor, hence saying mother's kiss can heal a wound. Esophagus is a muscular tube which connects mouth, or more precisely pharynx, to the stomach. This tube is about 25 centimeters or 10-inch long and is located posterior to the trachea. In other words, it is in the back of trachea, and it remains collapsed in the absence of food. The upper esophageal sphincter is a valve which regulates movement of food from mouth to the esophagus. The upper 2/3 of esophagus is composed of smooth and skeletal muscle, and towards the bottom of esophagus, the skeletal muscle fades out. These muscles contribute to rhythmic waves of peristalsis, which propel bolus of food to the stomach. Esophagus, like mouth, also secretes mucus, which lubricates the food, making it easy to move down. The lower esophageal sphincter allows food to enter the stomach. Once food enters the stomach, the valve of lower esophageal sphincter closes. Incomplete contraction or closing of the sphincter causes stomach content to back up into the esophagus, leading to heartburn and even gastroesophageal reflux disease. Stomach is a highly active organ, as peristalsis, which is simply contraction of stomach muscles, contribute to mechanical digestion of the food. Stretching of the stomach, on the other hand, allows stomach to collect and temporarily hold food until it is digested. The environment of the stomach is highly acidic, specifically ranging from pH 1.5 to pH 3.5. The acidity of the stomach is due to hydrochloric acid, which is secreted by parietal cells of the stomach. The acidity is important to kill microbes present in the food and to denature protein, making it more available for enzymatic digestion. As acidic environment inactivates certain enzymes, such as salivary amylase, it activates other enzymes, such as lingual lipase, enzyme which initiate triglyceride digestion in the stomach. Acidic environment also converts inactive form of pepsinogen to its active form called pepsin, enzyme which initiate protein digestion in the stomach. Because the acidic pH is highly corrosive to the stomach mucus cells secrete bicarbonate-rich alkaline mucus, which lines the stomach, protecting it from acidity, as well as self-digestion by gastric enzymes. Aside from producing hydrochloric acid, parietal cells also make intrinsic factor, a transporter glycoprotein necessary for the absorption of vitamin B12 in the small intestine. Vitamin B12 is extremely important for proper neurological function and production of red blood cells. Therefore, people who undergo gastrectomy, in other words, stomach removal due to cancer, for example, must obtain vitamin B12 through injections. With exception to nutrients present in alcohol and aspirin, little if any absorption occurs in the stomach. From an anatomical point of view, stomach is composed of different parts. Cardia region is the point where the esophagus connects to the stomach. Fundus is a structure next to cardia in the shape of dome, therefore it is slightly elevated above the cardia. Body of the stomach is located below fundus and it is the main part of the stomach. Pylorus is right underneath the body and connects stomach to the duodenum of small intestine. Right under pylorus is pyloric sphincter, which regulates food movement from the stomach to the small intestine. Empty stomach is about the size of your fist. It is deflated inward, and its mucosa and submucosa create folds called ruga. However, when you consume food, stomach can stretch about 75 times of its empty volume, allowing it to fit 4 liters of food and fluid. Small intestine is a coiled tube that measures about 3 meters or 10 feet in living person. However, it can be twice as long in cadaver due to loss of muscle tone. It is pretty thin in diameter, about 2.5 centimeter or 1 inch, hence its name small. There are 3 main parts to the small intestine. The first 25 centimeters of the small intestine is called duodenum. It begins at the pyloric sphincter, which regulates entry of acidic chyme from the stomach. To protect the small intestine from the acidity in the chyme, duodenum walls are equipped with duodenal glands, which secrete bicarbonate-rich mucus that buffers the acidic chyme. Located in duodenal wall is also hepatopancreatic ampulla, which is where the bile duct and the main pancreatic duct join. This ampulla opens into duodenum through major hepatopancreatic sphincter, which regulates entry of pancreatic juice from the pancreas and bile from the liver. Both bile and pancreatic juice help small intestine to complete chemical digestion of the food. The middle area of the small intestine is called duodenum, which measures about 0.9 meters or 3 feet in length. And the last part of the small intestine is called ileum and measures about 1.8 meters or 6 feet in length. Walls of small intestine is lined with villi, small vascularized projections that measure between 0.5 to 1 millimeter in length. Each villus is made up of epithelial cells, also called absorptive cells. These epithelial cells have microvilli projections measuring less than 1 micrometer in length. Both villi and microvilli function to increase surface area of the small intestine to maximize nutrient absorption across the epithelial cells into the bloodstream. Unlike other nutrients, lipids are absorbed across epithelial cells into lymph node before they can enter bloodstream. 90% of water is also absorbed from the small intestine through a process called osmosis. Large intestine is the terminal part of the GI tract. It runs from appendix to the anus. Although large intestine is about 1/2 the size of small intestine, the diameter is about 3 inches wide, hence the name large. The primary function of this organ is to complete absorption of nutrient and water. However, since residue of chyme that enters the large intestine contains only few nutrients except water, large intestine can be completely removed without significantly affecting digestive function. Large intestine also function to form and eliminate feces from the body. Large intestine is subdivided into 4 main areas: cecum, colon, rectum, and anus. The ileocecal valve, located at the opening between ileum of the small intestine and cecum of the large intestine, controls flow of chyme from the small intestine to the large intestine. Anal cavity is composed of 2 sphincters: the internal anal sphincter, made up of smooth muscle, and external anal sphincter, which is made up of skeletal muscle. Both muscles contribute to the elimination of feces. Although mechanical digestion continues in the large intestine, chemical digestion is strictly from bacterial flora in the lumen of the colon. Specifically, Saccharomyces fermentation is the process through which bacteria breaks down some of the remaining carbohydrates. As a result, hydrogen, carbon dioxide, and methane create gas in the colon. This bacteria also participates in the synthesis of vitamins, such as biotin and vitamin K. Bacterial flora in the large intestine is mainly non-pathogenic and causes no harm as long as it stays in the gut. Liver is the largest gland in the body. It lies inferior to the diaphragm on the right side of the abdominal cavity. Liver is divided into two primary lobes, a large right lobe and smaller left lobe. Right underneath, there is porta hepatis, an opening through which hepatic bile duct, hepatic artery, and hepatic portal vein run. The function of hepatic artery is to deliver oxygen to the liver. In other words, it delivers oxygenated blood to the liver, whereas hepatic portal vein is low in oxygen, therefore we refer to it as deoxygenated. It delivers nutrients, drugs, and toxins absorbed from the small intestine for processing by liver cells called hepatocytes. Once nutrients are processed, hepatocytes either store them or release them to the sinusoid, a space filled with fenestrated capillaries, which then drain blood with nutrients into central vein and then through the hepatic vein to the inferior vena cava. Hepatocytes also participate in bile secretion. Bile is a yellow-brown or yellow-green alkaline solution with pH between 7.6 to 8.6. It is composed of water, bile salts, bile pigment called bilirubin, phospholipids, electrolytes, cholesterol, and triglycerides. Bile function to emulsify lipids in the small intestine. In other words, bile helps to break down large lipid globules into smaller lipid globules to increase surface area available for lipid digesting enzymes, such as pancreatic lipase. Once bile is formed, it is secreted into the bile ductules and then into bile ducts. Bile ducts then form right and left hepatic duct, which merge to exit the liver as common hepatic duct. Common hepatic duct then joins with cystic duct from gallbladder, forming common bile duct through which the bile enters small intestine. Gallbladder is a muscular sac located posterior to the right lobe of the liver. It functions to concentrate and store bile as not all of it enters duodenum directly from the liver. Gallbladder contracts and releases stored bile into the duodenum of the small intestine after the meal. Pancreas lies behind the stomach and its head is nested into the duodenum of the small intestine. It performs exocrine and endocrine function. An exocrine function is to secrete enzyme-rich pancreatic juice from the acinar cells into pancreatic duct, which then merges with the common pancreatic duct before entering duodenum through the hepatopancreatic ampulla. The pancreatic juice is a clear substance composed of water, salts, sodium bicarbonate, and digestive enzymes. Sodium bicarbonate creates alkaline environment with pH between 7.1 to 8.2, which buffers the acidic chyme entering the small intestine from the stomach. Alkaline environment also inactivates pepsin enzyme present in the chyme and creates an optimal environment for the activity of pH-sensitive digestive enzymes in the small intestine, specifically activation of enzyme trypsin from trypsinogen. If this enzyme was secreted by pancreas in its active form, then it would digest the pancreas. Other enzymes secreted by pancreas, such as pancreatic amylase, pancreatic lipase, and nuclease, is secreted from the pancreas in its active form, since they do not attack the pancreas as do protein-digesting enzymes. An endocrine function of the pancreas is to produce hormones and secrete them into the bloodstream. Specifically, endocrine cells called islets of Langerhans produce hormones such as insulin and glucagon.